So our first speaker is James Krieger. James's research focuses on computational analysis of protein dynamics and signaling. James did his PhD in molecular neurobiology at the LMB with Dr. Ingo Krieger, and also at the University of Cambridge. And he was studying ionotropic glutamate receptors using a wide range of techniques like confocal and super resolution light microscopy and computational biophysics of protein dynamics. In 2017, James moved as a postdoc to the lab of Yvette Bachar at the University of Pittsburgh. And there he continued working on glutamate receptors and other proteins, and was a key player in the development of pipelines for protein dynamics analysis using Python packages, including ProDy, which he will discuss today in his talk. And also James will soon join the Jose Maria Carasso and Carlos Oscar Susano in Madrid as a Marie Curie Fellow. So congratulations, James. So yeah, looking forward to your talk.
So thank you, Maya, for the introduction, and thank you to the organizers for selecting my talk and for putting on this great meeting. So I'm going to talk today about some recent developments that we made into the Python package for protein dynamics, focusing on large-scale analyses and the CryoDy pipeline for cryo-EM maps. And this is the work that I recently did in Yvette Bahar's lab in the University of Pittsburgh during my postdoc. So seeing as it's quite a short talk, there's just these few parts. I'm going to introduce ProDy and the elastic network models and normal mode analysis that are the main theoretical underpinnings of it, and then talk briefly about the Scienti pipeline for signature dynamics of protein families and the CryoDy pipeline for dynamics in cryo-EM. So the protein dynamics that I'm talking about is mainly global collective motions that's happening on the microsecond, millisecond timescales, as you can see in these movies. And these can be happening just naturally as a result of thermal fluctuations, or they can be activated or modulated by various events such as ligand or protein binding or post-translational modification or some mutations or collisions. And we've seen in various studies and talks that these kind of dynamics are important for cryo-EM, and we need to model them better in order to further understand our proteins and how they're working. So ProDy is a Python package that we can use to build pipelines to understand protein dynamics using two main types of approaches. One of them is to build a physical model and perform computational analyses. These are elastic network models, and we calculate normal modes. And the other way is we take an ensemble of structures and we align them, and we can perform some kind of dimensionality reduction, such as PCA, on this ensemble. And we also get similar kinds of modes of motion, which we can then visualize and quantitatively compare. And the user can input various different types of information, such as a PDB ID or a sequence, and there's many tools for extracting these structures. And now we have ProDy is in version 2.0 we recently published in Bioinformatics, and there's more than 2 million downloads across the globe. So the idea, what we do here is, rather than take a all atomic model which has a lot of detailed interactions and takes a long time to perform calculations and needs molecular dynamic simulations in water. We simplify the models down substantially so that you get a network of beads and springs, which you can see like this, and they're much more coarse grained. So you might have one bead for every amino acid residue, or maybe even a higher level of coarse graining. And this allows us to perform much more efficient and robust dynamic calculations simply by connecting the beads with uniform springs within a cutoff distance. And these, because they have such a simple kind of equation, suitable to normal mode analysis. And this example that I'm showing you, you have for the most common elastic network model, which is anisotropic network model, ANM, where we have the potential is a harmonic spring potential with a step function that defines the cutoff. And this allows us to easily build the Hessian matrix of second differentials, where each of the 3x3 sub-elements is describing the motions of the pairs of residues, how they're interacting with each other. And this matrix then dominates the dynamics because we're assuming that our structure is an energy potential minimum. So this allows us then to perform normal mode analysis where you decompose the Hessian matrix with eigenvector decomposition, and we obtain these kinds of global motions. And these are the most collective and most energetically favorable motions, and they have a ranking so that each one becomes successively less collective and less energetically favorable. And while this ranking is maybe not exactly precise and you don't always know that the first mode is the most important, it's usually that one of the first few can be clearly linked to biologically relevant transitions, and this method's seen wide use over the years, including in flexible splitting, as well as comparisons with cryo-EM data, such as this StructMap and HemDeMap methods of Carlos Oscar-Solzano and John Edge. So the idea was now to extend these methods further, and ProDy provides you with quite a toolkit for building these methods into analyzing various different things, including allosteric signaling with methods such as perturbation response scanning, and recently essential site scanning analysis, as well as modeling effects of environments, including membranes. And as I mentioned before, building ensembles and principal component analysis. And in particular, we now have these two new pipelines, Scienti and CryoDy, for ensemble normal mode analysis for comparing the modes of many structures and for cryo-EM maps. And also, I'll briefly mention there's some E&M guided simulation methods now available in ProDy as well, including adaptive A&M and cluster E&M. So first, I'm going to talk briefly about Scienti, which will be important for understanding the differences in dynamics between related structures. So you can see here, there's two main parts of this pipeline. The first part is an integration with many different methods for extracting homologous or related protein structures. And we can then build these ensembles and calculate normal modes from each of them and match them up. And the second part is then a new set of analysis tools, which allows you to compare and find signature properties, which are the means and standard deviations of the mode shape, square fluctuations, and cross correlations. And you can also then perform comparisons of different groups of proteins and compare their dynamics in the tree-based kind of view, like you can with sequence and structure. So here's a brief example of some results on some transporters. So you can see in this case, these are modes from the Gaussian network model. So they show residues that move in opposite directions, and there are clearly regions that are more conserved, but the overall mode shape in dark blue, the average is clearly well defined in all the cases, and we can compare for representative members and also for the cross correlations or covariance that we see that there are strong signals of regions that can move together in opposite directions across many of these proteins. And here's an example for the periplasmic binding proteins, which have an important role in signaling in various receptors. And there's some conserved motions across all of them, which you can use to classify the proteins by their different functional states. And this classification is similar to one that you might get from structure using RMSD or PCA as well. And it gives you quite a different kind of information to the information that you might get from looking at sequence alone, where rather than seeing these different states, we're more separating bacterial or eukaryotic families of proteins instead. So clearly, this method is allowing us to differentiate the dynamics of related proteins and relate different structures of the same protein, which could be important for understanding conformational landscapes and how they vary across different conformations. So now I'll move on to the CryoDy part. So this pipeline, the main innovation is that we've combined a pseudo-atom fitting method into ProDy, which allows us to fit feeds into cryo-EM maps. And this allows us to then have an even higher level of coarse graining, but it's good for large structures, which are nowadays often coming out of cryo-EM. And this allows us to then integrate these with the other kinds of analyses that are available in ProDy, including normal mode analysis, allosteric signaling. In this case, this is a hitting time analysis, and PCA and these E&M guided simulation transitions, such as adaptive A&M. And together, this allows us to quantify different properties of the protein. And we also have a domain decomposition method, which allows you to interpret which feeds belong to which parts of the structure as well. So the pseudo-atom fitting method that we've employed is called the TopologyRepresenting Network algorithm, which has been used by a number of groups and has shown good success for reproducing structure and dynamics of proteins. And the way it works is you treat the cryo-EM map as a probability distribution, and you sample node positions, which initially they're not necessarily very good, but then there's a self-organizing neural network that updates the positions of these nodes. And we have some parameters for affecting how they interact with each other and the convergence of the algorithm. And this has been described as a neural gas network because of the way that the neighboring nodes can interact with each other. And as you can see in this figure, as you go through the algorithm, you gradually end up with something that quite well fits the topology of your density. So we applied this to the chaperone Intrinsic CCT, and this allowed us to identify various different kinds of modes of motion that are important for closing the subunits of this two-ring architecture. And together with the domain decomposition through multiple steps, we were able to then identify the different subunits and see state-dependent motions that were important for the allosteric cycle. And I should note that in this case, we fitted 3,000 beads to a structure of CCT that had about 11 angstroms resolution. And this allows us to do better, maybe even than the low quality models that were initially fitted to the structure. And because it's only 3 residues per bead, this is much more efficient than using the atomic structures for standard A&M calculations. As I mentioned, we were also able to use this for E&M guided simulations, and the way we did that was by creating a nearest neighbor approach for mapping the beads against the residues in a close by structure. And we're of course working on other methods to do this more efficiently, but this was already sufficient to allow us to compare this with other structures and generate this trajectory that you can see closing the ring, and also to explore the conformational space with principal component analysis. And also we were able to use a step of the CryoDy sampling algorithm to sample accessible conformants related to the same conformational state. So in summary, I've told you about ProDy, which is application programming interface in Python for protein dynamics analysis using elastic network models, normal mode analysis, and PCA of experimental structure ensembles. We now have ProDy version 2 with many features, some of which I touched upon, and you can see more details on the website, including useful tutorials. And I particularly told you about the Scienti pipeline, which allows ensemble normal mode analysis and reveals and classifies signature dynamics of related structures, as well as the cryo-EM dynamics pipeline CryoDy, which uses the TRN algorithm for fitting pseudoatoms and can be combined with adaptive A&M and ClassD&M simulations and domain decomposition. So I'd like to thank the members of the Bahar Lab in which I did this work, and particularly those who listed here, as well as my collaborators and future mentors, Jose Maria Carrazzo and Carlos Oscar Solzano. And thank you everyone for listening. And now I'm happy to take questions, and seeing as that there's maybe not so much time, people could also ask me more detailed questions by email if they'd like.
Thank you very much, James. That was very nice. We have a couple of questions in the Q&A. The first question is from Katsu Murakami. CryoDy only works for EMF. They're asking, can it work for X-ray map as well? That's the first question.
We haven't tried it. I guess in principle it could do, but there may not be such a big advantage to doing that, as X-ray crystallography tends to give you a smaller structure where you're not going to necessarily run into the same kind of issues. We also have generally higher resolution, so we can trust the models that we get out and just use the standard elastic network models using a C alpha residue or maybe every few C alpha atoms for the close training instead.
So does that mean, actually that leads me to my question, does that mean that CryoDy is particularly useful for the lower resolution range rather than, or medium resolution rather than better than 5 angstrom, say?
Yeah, I think so.
Okay. And if you took pseudo-atomic models for let's say between 5 to 10 angstrom, and then run the normal mode analysis and compare to what you get from CryoDy, would you get similar results? Did you try that?
No, but I guess so. Usually the normal mode analysis, especially for these global collective modes, is quite robust against the kind of model that's used, and not sensitive to these kinds of details, so I expected it would be. We also did compare modes from CryoDy against residue scale elastic network models, and we found that then the modes were matching very well. We tried also, in addition to the standard A&M, some different forms with an exponential or inverse square distance relationships, and it also reproduced the dynamics fairly similarly as well.
Okay, great. Okay, last question quickly, and then you will answer the rest, I guess, in the Q&A. Great talk, James. This is from Yang Zhang. Do you think one could combine Scienti and CryoDy to analyze the dynamics of ensemble generated by cryo-EM to classify conformers?
So it is something that I'm definitely thinking about. I don't know to what extent it would really be helpful or not yet. But I guess in principle, if we can find some kind of way of relating the pseudoatoms in different structures from cryo-EM with each other, we could use it to classify the structure and dynamics of these conformers and create a conformational landscape map in the same way that we usually do with atomic structure.
Okay, thanks a lot, James, again for a wonderful talk. And we have to move on, unfortunately.